Dr. Silberberg earned his PhD in neurophysiology from the Hebrew University in Jerusalem. He has a very strong background, which includes investigating functions and physiological roles of ion channels. He's currently with the NIH National Institute of Neurological Disorders and Stroke, NINDS, as a program director focusing on the molecular structure, function, and regulation of ion channels and transporters. He's also the director for research quality, leading efforts to increase the excellence of science and the completeness of research reporting. And with that, I'll turn the presentation over to him.
Thank you, Kat. Can you all hear me?
Yes.
Is it good? Okay,
Yep.
Great. So I'm going to go to share my screen here. Here's my screen, and I'm going to just enlarge that, and I think we're good. Is that right?
Yes, yep, there might be a little bit of a delay. So, yep,
Perfect.
you're full screen now. Perfect.
Okay, thank you. And thank you all for joining me in this virtual meeting. I would like to say before I even start that I have a disclaimer to make, and that is that opinions I will voice are not official opinions of the National Institute of Health. And as you will find out, I have lots of opinions. So in 2014, the director of NIH, Dr. Francis Collins, and his principal deputy director, Larry Tabak, published this commentary in Nature titled NIH Plans to Enhance Reproducibility. So what I want to do today is to tell you what led to this commentary and what has transpired. So over the past 15 years, as I'm sure you all know, there have been many...
Shai,
yes.
I'm sorry to interrupt, but your slides are not changing.
Okay, sorry about that. Yeah. So I told you that I have a disclaimer and that NIH published this commentary, and I'm going to tell you what led to this and what has transpired. And I was told that you can't see me when I had this slide up, saying that there have been many papers in the last 15 years suggesting that there are issues with the quality and transparency of preclinical research. And give you just one example. There was this paper by Bayer Healthcare published in 2011 where they reported that 2/3 of studies that they initiated in the company, it's 43 out of 67 studies, that were based on findings that were published by the scientific academic community, they could not reproduce. So they got inconsistent results. And it was papers such as this that led to this term, the reproducibility crisis. I personally do not like this term because there can be many good reasons why results can't be reproduced, including that the person who's trying to reproduce it is not doing things properly. And it's certainly not a crisis because this has been going on for quite a while. So instead, I'd like to tell you what I believe are the main issues why we are having problems with getting similar results to others and in advancing science. And they are what I call bias, which is human nature, lack of transparency, and the element of chance. So I'll start with human nature. Exactly 400 years ago, Francis Bacon in the New Organon wrote that once a man's understanding has settled on something, it draws everything else also to support and agree with it. He goes on to say that it is an innate and constant mistake in the human understanding to be much more moved and excited by affirmatives than by negatives. Now, he doesn't say what he means by understanding. I mean, maybe that was in Old English, they knew what they were talking about. But we have a pretty good idea where it is, and I think it was perfectly described by the comedian Emo Phillips, who said that I used to think that the brain was the most wonderful organ in my body. Then I understood, then I realized who was telling me this. So clearly our understanding is in the brain, and what Francis Bacon, Emo Phillips, and many others are talking about are unconscious biases. And I'd like to give you just one kind of simple example of unconscious bias. Please move to the next slide.
Let me just remove the...
There we go, okay? So what you can see here are two tables, a drawing of two tables. I labeled one side of each table with a W, suggesting that that's the width of the table. And I'm going to ask a stupid question, and I'm not going to ask you to answer because I've given this talk many, many times, and I know what the vast majority of people are going to say. The question is, which table is wider? And almost everyone, including myself, would say that the table on the right is wider, right? So if you can click once, please. What you can see, I covered the table on the left with a rectangle. And now if you kind of slowly click a few times, you'll see that we are moving this rectangle over. Can you please do that? Okay, keep going, keep going. One more time, there we go. And you can see that they're exactly the same size. And if you can click one more time, even though we know that they are exactly the same shape, we look at this and we're still convinced that the table on the right is wider. Now, why do I give this stupid example? Well, because if you think about it, think how many things we do as scientists that depend on our visual system. If it's behavioral studies that we're looking and trying to evaluate how the animal is behaving, if it's functional MRI, if it's Western blots, if it's immunohistochemistry, all these things we assume that our brain is telling us what we're actually seeing, where in matter of fact, it might not. Now, there are many kinds of biases, if I could please have the next slide. And it's kind of hard to decide how to define biases. And I like the definition given by David Ranzerhoff. He said that the reliability of a study is determined by the investigator's choices about critical details of research design and conduct. In other words, it is our responsibility as investigators to make sure that we are minimizing unconscious biases, okay? To design the experiments appropriately. But if you click one more time, we'll see that he also said that bias is unintentional and unconscious. So how can we reconcile these two things? If it's unintentional and unconscious, how could we be responsible? And I think the solution he gives hits the nail on the head, if I can have the next part here. The process of addressing bias involves making everything equal during the design, conduct, and interpretation of a study, okay? And then reporting it in a transparent way. So how do we make everything equal? There are many things that have to go into that, and it's not that simple. But I think Natalie clearly explained to you why it's so important to blind and randomize in order to make everything equal between comparison groups. So Natalie already gave an example. I'll give you another example. How widespread is the reporting on blinding and randomization? Next slide, please. So this is an example, a study, again, done by Malcolm McLeod and his colleagues in Edinburgh. And what he's, I have to just got something on my screen here. What they did is they looked at animal studies in 4 different neurological disorders. And they looked for elements of potential bias. If I can have the next slide, please. We're going to focus just on multiple sclerosis because it had the largest number of papers, 1,117. And you can see that only 16% of the papers reported on whether they blinded the analysis. Only 9% reported on whether they randomized to comparison groups. And look at this number, only 2 out of the 1,117 papers reported on how they came up with sample size. Now, this is not unique to the neuroscience field. If I can have the next slide. In an earlier paper by Hakam and Rittermeyer, they looked at 7 obscure journals that you see listed there on the left for animal studies that had a large impact. And they defined that as papers that were each cited at least 500 times and that contributed to a human clinical trial. And as you can see, only 20% reported on blinding, only 14% reported on randomization. So all I've told you so far is that people don't report, but do they actually do it or not? Natalie touched on this, and I'm going to give you a different example that suggests that if they don't report, they also actually don't do it. If I can have the next slide, please. And this is a study from also again by the Edinburgh group. They looked at all the papers that examined the effect of the immunosuppressive drug FK506 in experimental stroke models. And what they did is for each study, they gave a quality score. So if the study reported on blinding, they got a point. If they reported on randomization, they got a point. If they reported on sample size calculation, they got a point. And they could accumulate up to 10 points. And they called that the quality of the study, and that's shown on the x-axis. As you can see, none of the studies got all 10 points. Now, the y-axis reports the efficacy of the drug as it was reported in each one of these studies. And I think it's quite obvious that the fewer parameters that were reported, the greater the apparent efficacy, suggesting that lack of reporting is associated with lack of practice, and that this lack of practice can lead to bias. So why is this happening? If I could please have the next slide. There are many possible contributing factors. I want to focus on one important one, which I call paper money. If you don't provide papers, you don't get the money. Okay? If you want grant support, you've got to publish. And you have to publish relatively frequently, preferentially in high impact journals, which have a high impact factor, whatever that means. And for that, the study has to be novel, innovative, and significant. And this whole cycle is on average, at least in the US, about 4 years. So how could you possibly publish a few such great papers in just 4 years? You know, it's really not realistic. So what happens is investigators tend to rush studies to publication. Okay, they publish papers that could have benefited perhaps from a couple more controls, experiments that perhaps would have used a larger sample size, or maybe a couple more experiments that try to disprove the hypothesis rather to show that it's right. Okay? So these all are contributing factors. And I want to give you an example, a story. Natalie touched on this example, but I'll give it from a different perspective. I can have the next slide, please. It relates to the devastating disease ALS. So as you know, it's a neurodegenerative disease. Within 5 years, typically leads to death. And in the 1980s, investigators succeeded in putting together or creating an animal model that was used to study ALS. It's a mouse model that overexpresses the human superoxide dismutase 1 gene. And the phenotypes have many things in common with human ALS. And if I have the next part of the slide here, please. In 2002, a couple of groups reported that the antibiotic minocycline significantly enhanced the lifespan of the SOD1 mice. Because ALS is so devastating, because the results were so significant, and because anyone can get hold of minocycline, NIH decided within 1 year to fund a clinical trial. And if I can have the next slide, please. In 2003, a placebo-controlled trial was funded and initiated. 412 patients were treated for 9 months. And the next click of the mouse, please. The results were published in 2007, and there was no beneficial effect for minocycline. So why am I telling you this story? I mean, there's no reason to think that what works in animals is necessarily going to translate to humans. I mention it because of the next slide. A year after the clinical trial was concluded, there was a paper from the ALS Therapy Development Institute in which they report on 4 years of work where they carefully evaluated more than 70 drugs in the ALS, in the SOD1 mouse model. And they used really, really rigorous procedures which allowed them to detect a statistical significant difference between male and female longevity on the order of a few days. However, next click, they found absolutely no statistical significant effect of any of these 70 compounds, many of which that were previously reported to be efficacious, one of which was minocycline. Next slide, please. They go on in the paper to write that the majority of published effects are most likely measurements of noise in the distribution of survival means as opposed to actual drug effect. I think this is a very polite way of just saying, if I have next click, please, that these results are simply by chance, okay? Now that's a pretty audacious statement. So how did they come up with it? Well, because as Natalie already told you, they had so many experiments that they did. They had actually 2,241 control animals that they just lived out their lives in the cages, okay? And they knew everything about them, when they were born and when they died, what sex they had, what litter they came from, how many copies of the transgenes, what was the cause of death. They had all that information and they put it into a database. And then they could run this really cool in silico experiment. They could randomly select animals from this database, put it in two separate groups and ask the question, what is the probability of seeing a difference in their life expectancy between the two groups, which by definition would be by chance because these are all control animals. And they defined significant as a 5% difference in life expectancy. Now, when they adjusted the groups so that they were, properly adjusted for sex and litter and cause of death, and number of transgene copies, et cetera, they got the following results. If I can have the next slide, please. And Natalie touched upon this, but I'm going to give a different angle to it. Can I have the next slide, please? Thank you. No, one back, please. Okay, so if they had 4 animals per group and they adjusted for everything, they had about a 30% chance of seeing a so-called a significant effect, which by definition is by chance, which means it's a misleading result. Now, some of you might ask, who in their right mind would use only 4 animals per group? So I just want to remind you that the study by Hakam and Rittermeyer that looked at those 7 obscure journals, half the papers they looked at had 5 or less animals per group, okay? Next click. If they had 10 animals per group, now they improved it to about only 10% misleading results. So let's say a right answer 9 out of 10 times. And the next click, what they conclude in the paper is that if you really want to minimize the chance that the probability that you'll get a chance result, you have to have 12 animals of each sex per group. That's when you're studying the SOD1 mouse model and looking at longevity, okay? I mean, obviously this does not apply to every study. So with that information, let's go back and look at those papers that led to the human clinical trial. Next slide, please. The first paper was published in Nature. What you see here is the key figure. It's a survival plot. The dashed lines is the control animals. The solid line are the animals treated with minocycline. You can see that in this study, they used 10 animals per group, no mention of the sex. And we know that if they didn't adjust for the sex there, we would expect differences because the animals live longer, different length, females live a little longer. There's no mention of randomization, no mention of blinding. A second paper published that same year, next please, had kind of similar results. The control group is on the left, 2 concentrations of minocycline on the right. Here they use 7 females per group, no mention of randomization. They say that the experimenter was blinded to the treatment protocol, but as Natalie pointed out, you have to blind yourself throughout, the whole stages, including the randomization stage, the conduct of the experiment, and the analysis. And it's not clear what they did here. So because we know that when the study was done carefully, there's no effect on minocycline. Next, please. I think it would be fair to conclude that the results, no, you want too much. That the results from these early studies were either due to small sample size and or unconscious biases. And it's kind of tragic to think that patients were exposed to a treatment that they should not have been. Next slide, please. So taking a holistic view on what I just told you, clearly there's lack of transparency in reporting. There's deficiency in experimental procedures that could lead to risk of bias. And we all know that there are perverse incentives like the paper money that could lead people to, or scientists to rush studies to publication. So how do we address this? Next, please. To improve transparency, we could try to change the review of manuscripts and grants. Next, oh, this is so slow. To address deficiency in experimental procedures, obviously we have to increase awareness and to improve education. And for perverse incentives, we need to change the culture. If I can see that, please. And that's, of course, a much more difficult thing to do. So recognizing this, next slide, NINDS organized a workshop in 2012 with editors of prominent journals, reviewers, investigators, and funders that resulted in the paper you see here. It's commonly known as the Landis et al. Paper. And the key recommendation of this paper, next please, was that when you're doing animal studies, at a minimum, you should report on sample size estimation, on randomization, blinding, and on handling of data, which included inclusion-exclusion criteria, and so on. This is the bare minimum that we kind of all agreed upon. And I think because we had such a limited number of items, this paper actually had quite a significant impact. And just to illustrate this, if I can have the next slide, within 8 months, Nature Publishing Group made quite significant changes to their review process, including adding a checklist that included the 4 items that are now commonly called the Landis 4. NIH 2 years later held another workshop with journal editors, and the number of journals that have made changes is continuing to increase. Next slide, please. So what did NIH do in order to improve the review of grants? So NIH in 2016 introduces enhancement to grant review that were modified again in 2019. And they have 4 pillars. If I can see the first pillar, please. The first one is rigor of the prior research. Initially, they called it scientific premise. Most people didn't understand what it meant, so they finally changed that. And if I just quote from here, NIH applicants to describe the general strengths and weaknesses in the rigor of the prior research that serves as the key support for the proposed project. Not all the literature. Not, if you can say, oxygen is important for survival. You don't have to go and check all those papers. But any paper or preliminary result that you use in support, the key support for your application, you've got to describe whether there are any issues with the potential rigor of that study. And if you identify gaps, the investigator needs to say what they're going to do to address those gaps. Okay, so that talks about what supports the application, and that's to assure that NIH is not going to fund a house of cards. Next, please, is the rigor of the proposed research. Here, the investigator has to describe measures taken to minimize unconscious biases and chance observation. So obviously, they have to talk about blinding, randomization, sample size estimation, etc. Okay, next. The third is biological variables, such as sex. Because it said such as sex, that's where all the attention went to. But in reality, biological variables covers many aspects. For example, let's say you're a stroke researcher. Does it make sense to work on 6-week-old rats that are normotensive, or should you be doing your studies on aged animals that have hypertension, okay, for example? And the final pillar is authentication of resources. We know that there are lots of issues with the quality of antibodies, that 30% of cell lines are probably misidentified. What are you going to do to authenticate your resources? So these are the things that NIH did as far as improving the transparency in grant review. If I can move on, please. So there's next, please. So we addressed, transparency. Obviously, there's a lot more that needs to be done. But what do we do about, education and changing the culture? So to this end, if I can have the next slide, in 2018, October 2018, NINDS held a workshop with prominent neuroscientists and trainees, scholars of education and science communication, and educational platform developers. And I don't have in this picture Natalie, but Natalie was also at this meeting. And we had very interesting discussions on how to, could we create a platform for instilling the principles of rigorous research that institutions can use to better train people, because we know that most institutions actually don't teach these things. And the recommendations from this workshop, next slide please, were recently published in this eLife paper. And the bottom line was this. I mean, it was clear during the discussions that unless we change the culture simultaneously with providing an educational platform, we're not going to make much progress. So we call for the establishment of communities of rigor champions that will help change the culture. It means a grassroots effort to change the culture in favor of rigor instead of glitter. And these communities will also be involved in the development, evaluation, and adoption of the educational platform. So if you're interested in joining as a rigor champion, if you can click the mouse again, please, you can turn to Rigor Champions at nih.gov and sign on. Unfortunately, our paper came out in early March, just as COVID-19 hit. So traction is still being relatively slow, but we hope it'll pick up soon. And next, please. We also published a request for information for input on the specific parameters of the educational platform we are proposing. We would love to hear from everyone what they think, what should be added, how it should be added, how best to do things. If you're interested, you can just copy the notice number at the top or go to the link at the bottom, and you will be taken to the request for information. So I'd like to end with my very last slide. It'll take a village to really improve rigor and transparency. We have to have the funders, the journals, the institutions, and perhaps above all, the investigators to really work together to improve the quality of science. And with that, I'll end, and thank you for your attention. I apologize for the technical difficulties, and I'll be happy to take questions.
Thank you so much. We have two questions for you. Let me read it really quick. Okay, the first one is, can you offer any tips on getting university to adopt the rigor changes, particularly as an AWIC member facing PIs that do not want to change or take on more work?
Institutions, so yeah, this is a difficult question, and I have to make sure, this is not my NIH hat, okay? I think, again, without my NIH hat, I think that the institutions are going to do what they think will benefit them as far as getting funds. So at some level, the more attention that will be paid on grant review to the quality of the proposed research and not how many papers the investigator published in a single word journal, the institutions will gradually understand that they need to do more. So I think it has to be a combination of carrots and sticks. For example, in our training grants, they are required to teach the principles of rigorous research, and they don't have a course to do it. And it doesn't surprise me because it takes a tremendous amount of effort to try to put a course like that together. Not only effort, you have to have the know-how, I said expertise, the time. So if we put together this a resource that will be freely available, modular, upgradable, easy to use, and it's like a one-stop shopping. You come and you pick what you want from that to put together your course. I think that would greatly reduce the energy barrier to improving education. On top of that, there are lots of things that the institutions can do, and this has to come from the investigators. And we see that there are lots of things like that going on. For example, at Johns Hopkins University, there's an initiative to try to improve the rigor of research. And this didn't come from the leaders. It came, from a grassroots effort. The same is true in Germany. There are examples like that. And in the UK, they've got this rigor initiative, which is kind of like communities of champions. Just we're thinking more globally, not only in the UK. So I think that the pressure has to come primarily bottom up, but we have to also identify the champions among the leaders in the institutions that are on board and that are willing to help to make change. You said
That's
there was
great.
another question. Yeah.
Yes, actually, I think that answered the second question, which was, if a change in culture is needed, does that also necessitate a change in the pressures of publish or perish and the demand for so much preliminary data in grant applications?
So I'm not sure, you know, so first of all, there are different kinds of grants, right? So there's the grants that you don't need a lot of preliminary data because of the nature of them. You know, if it's an exploratory, you know, at least in the US, if you have an R21 that you're putting in, you know, you're not required to have that much preliminary data. You just have to have enough to convince the reviewers that you're capable of doing it and that it's a good thing to do. As far as you know, the number of publications and, you know, and how reviewers look at that, NIH has been trying to change that for some time now. For example, they changed the biosketch. So now investigators are required to list 5 projects for which they believe they've had significant contributions, to explain what their contribution was. The idea is that the reviewers will read and not only count. And you know, all these things will take a tremendous amount of time because the big problem is the reviewers are our peers. They are the same ones that feel the pressures at the university. They're the same ones who are publishing rush studies. They're the same ones who don't pay attention to rigor and blinding and I mean to blinding and randomization, etc. So to change that, that'll take time, but I think we're moving in the right direction.
I had a question, and any of you are welcome to answer this, but we focus a lot on the authors and the investigators and their role in reporting all of these components. But I wonder if there's also a space for some of the onus to be put on the reviewers, where they're expected to look at these checklists also and think about whether or not they're going to accept the paper based on what's reported.
So yes, I can't speak for all the journals, obviously, but my understanding is that the reviewers in many journals are required to look at the checklist. In fact, what Nature Publishing Group did, which was, I think, a good way to help reviewers and also make sure that the authors are paying attention, is to say, not did you do this or did you report on that, but tell us where it is mentioned in the manuscript, okay? And that, of course, is a big difference. I can tell you, I mean, there's been a study that was done that already shows that the changes that Nature Publishing Group has done has made a difference. There are improvements in reporting. Now, it's not an important thing that he did, and I think, you know, at this point, it's legitimate, and I mean, it should always be actually. The most important thing is to improve transparency. So it's okay if you say, I didn't blind these experiments. It's, you know, maybe it's not okay as far as the science goes, but as long as you tell us, the reviewers are in a better place to say, okay, do I think I could believe these results or not? And the readers do as well, okay? So just having the transparency of what was done is a long way to, you know, maybe the minocycline study would not have gone forward. I say maybe because this was, you know, a long time ago. Maybe it would have not gone forward if the papers would have said this wasn't blinded and wasn't randomized.